Hello and welcome to the Science for Societal Progress podcast. In this episode, we talk about a new completely free and open non-profit type of journal, the role researchers should play in the dissemination of science to the public, and we revisit episode 11, where Elaine Pidon and I discussed GMOs. My co-host is Bart Geurten. I am your host, Dennis Eckmeyer, and you are listening to episode 14 of the Science for Societal Progress podcast. Before we jump in, let me make a brief announcement. We always welcome your questions, critique or suggestions. You can send us an email to info@scienceforprogress.eu, message us on social media @sci4progress on Facebook, Twitter and Instagram, or leave a video message on Skype for the account dennis.eckmeyer. A more direct way of getting into contact is our monthly member meeting on Discord, a voice and text chat service. To be invited to these meetings, you need to be a member on Patreon. In fact, if you like our work and want to help us sustain or even grow and improve Science for Progress, Patreon is the best way to do it. It is a service for membership-supported organizations, and just as in any society, club or association, you can choose your level of engagement yourself. But on Patreon, you can also choose your membership fees yourself. If your pledge exceeds a threshold, you enter a membership tier and you get some perks. So while you're listening to this episode, you can head over to www.patreon.com/sci4progress and check it out. How have you been, Bart?
It's so nice to be back here, in my own living room. Today my second student handed in her PhD thesis, Christina Kothal's.
Oh, awesome.
So she will defend on the 18th, and I'm pumped to see her work in conclusion.
Cool. Very glad to hear that. And she has a postdoc coming up?
Yeah, she's going to the University of Lund. I'm very proud of her. I think she's going to do very fine. Sometimes it's a bit sad to see them all go, but yeah. On the other hand, I'm happy that they all found very fast postdoc.
Are there new ones coming up?
I'm in this position where I just write grants to find new ways to hire people. And I have some very promising bachelor and master students at the moment, but I'm really afraid of like dragging them on without having found a clear position to put them on. So usually in this cases, heavy-hearted, I'm looking for other places to and colleagues where I can look out for them for a PhD position instead of making them wait for a position that I might never get.
Yeah, no, absolutely. Yeah.
But
yeah, I think that's better. I have had so many people who said, "Oh yeah, you can totally be my postdoc. I just have to get the money somehow." You can be my postdoc. I just have to win the lottery before. So,
fair enough.
Yeah, I'm being unfair. They meant good, right? They meant well, but it's just the reality that if the answer is I just need to get the money real quick, that's
no. So that's the reason why I then talk to them and ask them. Usually it works out.
Cool.
And today I gave the first lecture of the semester. I think I did a pretty nice job.
At least you're confident.
And I really like my voice, so
I will ask your students once I meet them.
oh, please don't. But you have great news too, don't you? You just turned 40.
Yes, I did. I did turn 40, which is funny because so the weird thing about it is I don't know. I never imagined me not having a job at 40. So yeah, I met a couple of PhD students from my previous lab, and we hung out, had some burgers.
So the funny thing is that I always forget that Dennis is older than me.
Yes, he keeps forgetting that.
I always think that he's like 3 or 4 years younger.
I started doing YouTube videos. So my first project, of course, is still the science advocacy. But since I don't have a job, I'm trying to make myself known as a professional. And I want to do some YouTube videos on neuroscience and scholarship. But the first video I put on is actually on how to be a good guest at a podcast. And it's focused on what you can do to improve your sound quality. And I put that online last week.
I see the pattern, man. It's okay. You could have said something.
What?
You could have said something.
About what?
You didn't need to make a YouTube video to tell me. All right.
You already knew. I sent you the PDF. Your sound quality is good. You're recording your sound yourself and everything. It's all good. Okay, so what else happened? There was news on Twitter. They founded a new so-called overlay journal. An overlay journal is a pretty new idea, actually. So you know that there are so-called preprint servers, which basically are archives where you can send your manuscript before it is peer-reviewed. And the reason these were invented was to accelerate the dissemination of scientific findings in the community. And now the overlay journals, they use this infrastructure, and really all they do is to organize peer review for articles on that archive. And the idea is to cut out pretty much everything that costs money. And so this journal is completely free. And because they are using the preprint servers, it's open access. And yeah. And it's called Neurons, Behavior, Data Analysis and Theory. And it's a neuroscience journal.
So is it instead of publishing, or is it surplus to publishing with a canonical publisher?
No, this is instead. This is a journal. And it's not a commercial journal. It's completely community-based. So all the work that is done is volunteer work, because all they do is provide peer review, and peer review is community service anyways, and hardly anybody is paid for that.
That's true, but they also don't generate income.
No, they don't.
How are they going to pay the people who make the journal impact factor then?
They won't.
I mean, otherwise they just have to count their citations and divide them by the number of articles.
Yeah, that would be...
That's preposterous.
Exactly. That's actually the main problem here. So they were asking what would prohibit you from submitting an article to us. And the main response to that, I think more than 80%. So that was a Twitter poll. They said it's the journal impact factor, because they have to look out for their careers. And first of all, a journal that's that young doesn't have a journal impact factor yet. And then, of course, they won't be paying for a bigger number. Yeah.
So what is needed now is some risk-friendly researchers that say we give this a shot and send something there.
Yes, exactly.
Is it already out there?
It's already out there, yes. You can look at it at nbdt.scholasticahq.com or at nbdt_journal on Twitter. So again, idealistically, I think this is actually a good idea, because you cut out all the money. And the money is the one big problem when we're talking about paywalling articles, and when we're talking about open access journals having an incentive to publish as much as possible, and they have an incentive to decrease peer review quality in order to publish more articles, because the authors pay for the article only if it's published, obviously.
Is there anything known about like their organizational body? Are they a club, or are they hosted by an institute, or?
No, they're just a group of people who do that. They're just a community. On the top level, there are 3 people who organize the whole thing. And then they have 130 editors. And then there is an undisclosed number of reviewers. You can actually go to their website and sign up as a reviewer through a form.
Do they already have published articles?
They have one so far. Yeah, well, they're really new. They announced this November 13, I think. So that's a week ago today.
So I'm waiting since 90 days on a review of a canonical publisher.
Jesus.
I'm not going to a second round of reviews, obviously, with these people, because after 90 days, I'm not going to do that. Then I also could send it to NBDT. Why not? Give it a try.
Yeah, I think that's a great idea. Okay, so let's move on. We had a monthly member meeting last Saturday, and we were talking about what is the role of the researchers themselves in science communication. And I remembered a YouTube video that was done by the famous German science communicator Mai Thi Nguyen-Kim. And she commented that, yes, there is a profession for translating science papers or scientific work. However, she says there are not enough science journalists to cover all the science. So all the scientists should, or she wants to force them, basically, to write a layman summary of every article that they write. And I feel like scientists and journalists try to throw the ball in the other party's field here. Because the journalists, science journalists, are supposedly the professionals to do that. And I feel like researchers, especially, they just have so many things to do already. And I feel like taking up more and more the work of other related professions needs to stop at some point.
I would agree that, for example, a university scientist has to fulfill many roles, like teacher, scientist, money acquisition, presentation, a lot of administration. Yeah. On the other hand, there's a need for justification in society. Like, people want us to be graspable in a way, so that they can afterwards say, okay, this is the reason why they did this thing.
Right.
And at a certain point I would say, I think I can do that. But I guess another person who never had a scientific background says he only thinks that he can do that.
Yeah, I agree with that.
If I try to explain the carbon filter to a layman, I guess I lose most people by the idea that there's a mathematical filter. That's not arrogance, it's just like, I have no idea how most things on a boat are called.
Right.
I find it already startling that they have starboard and backboard. People who listen to my lab slang are as lost as I am lost on a boat.
So Mai Thi Nguyen-Kim's argument would be that, oh, this is just another language that scientists already speak, the layperson's language, and we just have to translate it from science language or jargon to layperson's language.
I could try, and maybe this is the better thing that a journalist then can use, like a stepping stone to get to what I really want to say, maybe.
So the target for these summaries are not science journalists who maybe would have a chance to then understand it if it's still a little bit jargony, or if it makes too many assumptions about the education of the reader. Are lay people who didn't have science since high school? I mean, if you want to reach people who stopped having anything to do with science after 10th grade or something, then you really have to explain a lot of things, because they're not used to that, and they didn't go through 10 additional years of education at university to learn the language. So that's the reason we are using scientific terms, why we have our own terms, is because we're talking about things and concepts that don't exist in the world of the layperson. So we can't just translate it because there are no words for that. We have to explain what the words are to begin with.
Let's make a game out of it. You pick a paper and I pick a paper. And then we write a layman's term abstract of it.
Uh-huh.
about the size of a Journal of Experimental Biology abstract.
Okay. But we need to keep track of how long it takes, because that's one of the major issues here, right?
Okay, that's fair enough. And then we need some middle school pupils that have to read this in English and tell us if they understood anything and what they understood from this text. I guess we have some friends who have kids in that age, and they could record their responses for us.
Okay, let's try that.
I'll pick the meanest paper I can find.
Yeah, likewise. And don't forget, I'm the one saying it won't work anyways.
Wait. I picked the wrong tactic for that. There's something wrong going on here.
No, I will try my best, obviously. Okay. Yeah, we had this episode about GMOs. That was interesting, I thought.
Yeah, it wasn't very surprising to me, as I work with GMOs all day long. But then it's flies and not plants, and so I was pretty surprised how long genetic modification is going on in human history since like ages ago. And I can understand the frustration there is about people not understanding what actually genetic modification is.
I think a lot of people don't understand what is happening inside the plant when you do these traditional breeding methods. So just to repeat that, artificial selection is to look at the plant, and if the plant has some characteristic that is interesting, then you pick that plant and only breed that one. So then the next year, you only use seeds of that plant on your field. And in this way, you basically do what natural selection does, which is taking a random variation in the plant and then only allow that one variation that is beneficial to humans in this case to survive. And that is already a genetic modification. That is already quote unquote artificial and not natural. I was surprised how big these changes were, though. We had enormous changes in the genome of the plants over time, and we know that because we can compare them to wild plants that are still around and that we think were the original version of the plant. And they're straight up different species in many cases. They can't interbreed anymore.
I thought about was like if this is actually more than a twofold problem, because people are not so much interested in genetic modification itself, but in food that is genetically modified. In a way, I think most people are not aware that there are genes in any kind of food that you eat, because it's made from cells, and what else should be in there but genes to create those cells.
So that's the first point. And then there is that there are genes that cause the plants to have certain characteristics that are better than the original version of the gene. And then they select for that, or they do this crossbreeding, and it's all about getting this version of the gene into your main crop, so that you have high yield. And what the new methods do, and that was also surprising to me. I didn't know that they would go that far. There's the mutagenesis thing, where all they do is enhance the probability of the variation by introducing mutagens. That's what they're called. So if you change a gene, that's a mutation, and you can do that randomly across the whole genome using chemicals or using radiation, and then you hope for one of these variations to cause a beneficial characteristic. And they literally put radioactive sources into the field and see what happened in the 1970s. Today there are better ways to do it.
First of all, I have to remind you that this is not a laughable matter, as this is the way the turtles came about. And
what?
The Teenage Mutant Turtles came about this way.
That's not a laughable.
And they make up a large percentage of our childhood.
Yeah.
And on top of that, actually the same thing was done in Drosophila. And a lot of the canonical mutants that we use nowadays are from those radioactive tests. Mm-hmm. And they are really a mess to work with, because you have like one very prominent mutation that then is associated with all the phenotypes of that.
So the way it looks and the way it behaves.
Etc., etc. Exactly. Mm-hmm. But there's also what we call side hits. So there's this one big mutation that changed the eye color, let's say. Mm-hmm. But there's also other side hits on the same mutated genome that also make it, for example, slower. Huh. And then you don't know if, like, the gene that regulates eye color also regulates locomotion speed, or if that's a side hit. But yeah, I guess if we did this with Drosophila to find out how the genome in animals work, we did the same thing with plants. And if you get like your super plant or a Teenage Mutant Hero Turtle, then why not keep it? I think that's exactly those mutations that have something to do with radioactive mutagens or chemical mutagens scare people. Because if it's not good for yourself, and obviously radioactive radiation is not good for yourself, why is it good for my food? And do I get cancer from it? I think this is a lot of scary stuff to many people. And it's really, I think this is like coming back to our question from before, how do I communicate science? It's important to say that the mutant itself is, for example, not radioactive anymore.
Right.
A mutant is not a mutagen himself. Like, I can eat a mutated plant and nothing happens to me, as I digest, let's say, the naive form of the plant all the time. And that has had genes too. Eating a pea doesn't make me a pea human.
So it's like how you can become Spider-Man by being bitten by a radioactive spider. But if you're being bitten by the radioactive spider's non-radioactive offspring, you will not become Spider-Man.
Oh, this is so much more scientific than the turtles.
You started with the turtles, so I had to come up with something myself.
Yeah, totally right. So, but I think all these tales are so much more present in pop culture than the complicated theory about genes, mutagens, mutations, and how they propagate.
Let's say, in principle, you could have an accidental mutation that causes the plant to become toxic because it produces a new protein that could harm you.
Okay, fair enough. I accept that, but the chance of producing something toxic is much more higher with classical mutation, just by trial and error, like we do since the agricultural revolution, than with CRISPR-Cas, where I have like perfect control over what I do, or nearly perfect control.
Yeah, exactly. And that was kind of my point that I was trying to make in that episode, together with Elaine, that we have these older methods that were using these pretty random mutagens to cause a higher mutation rate in the plants, so you would get a higher variation, so you can have more plants to choose from and have a higher chance to find one that has a higher yield than the previous version, or another characteristic that you want to add to your main crop. These have all these side effects because it's random, it's all over the place, so you can have these problems. Now, the European Union court decided that those methods are safe because we have, I think, about 50 or 60 years of history using plants that were generated with these methods, and these plants have all been shown to be safe. And now we come to the point that you were making, that if you use a gene editing tool like CRISPR-Cas or transgenesis methods, which are actually still more used, where you transfer one gene into the plant that wasn't there before. It's a complete gene. The source of that gene is irrelevant. It could be anything. So that is way more controlled than if you do this random mutation and you have a smaller chance of having weird side effects. So that's what you were saying, right?
I mean, I understand also the argumentation of the court saying things that are safe for 50 years we trust. I totally disagree with the how's it called, verdict of the court, as I think that CRISPR-Cas is a much better method to create new plants. But I can understand the argumentation. The problem, though, is that it's not really scientifically driven. It's a decision based on a feeling for security and fear, and this is not, basically decisions made by these feelings are not very helpful or logical.
So yeah, exactly. So the logical thing would be to say, okay, so we have been using these things for several decades, and we now decided that we want to have regulations on this kind of manipulation in plants that we eat. I understand that in that case, because of historical reasons, you would go back and say, okay, we found these methods to be safe because we've been eating these plants forever, and nobody ever died from eating a GMO plant. So where it becomes illogical is when they say, okay, these are safe, and then with complete disregard of the mechanisms, they say, oh, but these new methods have not proven safe. Although, if you understand the mechanism, there is no way to assume that these new methods would be more dangerous than the older ones.
On the other hand, I think these judges also have to bear in mind how other parts of this gene technology are actually managed. And I don't know if they only put their verdict onto the logical part, the logical basis and arguments of CRISPR-Cas versus old mutation techniques, or if they also looked, for example, at how are these plants marketed. And that's a real problem.
Well, the verdict doesn't only talks about safety. They don't say, for example, oh, if you create new genes, should those be copyrighted? And these are other questions, but we don't even get to ask these questions and attack these questions if you ban the method just because you say, oh, it's not proven safe yet, although there is no reason to think it would be less safe than the methods that we continue to use.
Do agriculturists have to prove that CRISPR-Cas is safe, or does somebody else have to prove that it's unsafe?
So we have to prove that it's safe.
How do you do that anyways?
That's the huge question. The big problem that I have here is that they're looking at methods rather than the actual outcome. So, I mean, these are editing methods. Every time you use it, you may have a different outcome. It depends on what you're changing and what you're working on and which genes you are transferring or introducing. That has nothing to do with the method. These kind of things like new genes and stuff can show up in your plants by artificial selection, by crossbreeding, by mutagenesis in the old style, by gene editing, by all these methods. And they're not looking at the plant in the end and say, oh, we make a chemical analysis of this new crop, and if we don't find anything that's on the list, then we agree that it's safe to eat. Instead, what they're doing is, oh, there's this odd method. I don't trust the crop because of the method it was produced by. And that's, again, for me, something that makes very little sense. The main issue why people are against genes is really just because they're afraid of the technique. You said they might be thinking that the mutagens are still in the thing that you eat. Some just find it. Some might be afraid that side effects occur that make the plants toxic. Some will just find it unnatural. We are playing God if we take a gene from a bacterium and put it in a plant.
Ah, okay. I mean, but these people who are afraid of humankind taking God's power into their hands, did they ever look at pugs? Like, all these dog races are just human engineered.
And in the case of pugs, it's really not good. You should go Google pugs skull and then come back to me and say it's okay to like pugs, because I don't think what we did to those poor things is okay. The skull and the jaws don't fit together, really.
Oh my fucking God.
Oh, you just Googled it.
Oh God.
Should we breed dogs like that?
Would you agree to like keep pugs around if you can restore them to a more naturalistic like dog breed?
Well, then it's not a pug anymore. If you want to create a new breed through crossbreeding or something that is less effed up, then be my guest. But why not just take one of the hundreds of breeds that are not that bad?
Anyhow, okay, pugs are weird mutations.
Right. And we do things like that to our farm animals too, right? To stay in parallel to agriculture, to stay in with agriculture.
I think it's part of the hubris of modern society to say, on the one hand, we want very cheap food, and maybe we want to feed a very overcrowded population of people.
Well, we have to.
And on the other hand, we don't want to use the most modern available techniques. And if you talk to people about like how would they like their produce to be grown, they think about like this farmer from their children's book. Modern agriculture doesn't look like that anymore, and has to look differently to be able to produce enough food for everybody. Now, you could easily argument that there's not enough food going around for everybody, otherwise there wouldn't be hunger in the world, and that we have a large overproduction.
You have overproduction in Europe, and then Europeans sell their food for really cheap to countries in Africa and destroy the market there because the local farmers can't compete. And African countries made very restrictive laws against GMOs, but in several cases now, genetic modification of the modern type actually allows to rescue whole species. So that's one point Hélène made that I think is really important. We can use genetic modification now to restore some of the resilience that the wild plants have in our crops. And they lost it because we artificially selected only for high yield, and they lost some other properties on the way. And especially if we look at climate change and things like that, we want to make our crops more hardy so we don't have as much crop loss as we had this year, for example, with the wheat in Germany because of the drought.
What I found interesting is that seemingly all these big companies who are in crop design, like Bayer Crop Science, Monsanto, BASF, etc., that they don't use their influence and power to actually educate people.
Oh, they try to do that. But then the people who get paid by Monsanto to do education are being attacked for being shills for Monsanto. They just gave some travel grant to a guy who was going to give a talk to some people about genetic modification because he's a professor on this topic.
Like, there's no need for a professor of genetics to do that. Just tell people like it is. This is a mutagene. This is a mutant. Mutant is not dangerous anymore. The mutagene is dangerous to you as an organism. Don't come close to that. But you will never see that because this is not in your can. Corn is in your can. And it's the mutant, etc. Like all these like little parts of it. You don't become Spider-Man. Turtle in your fish tank still stays a turtle, etc., etc. That would be easy for these companies, and I don't understand why they don't do it, because then they could use it in more countries.
So what you're talking about would be like a series of TV ads explaining genetic modification?
Yeah. And they will have a marketing department. And instead of marketing the products, if you would market like the general idea of genetic modification and explain to people what's the difference between a mutagene, the mutant, the plant that they're eating, where a gene comes from, people would rather understand that. As soon as you understand something, it becomes less magical and less awful and awesome in a way.
Right.
Genes are still awesome.
It's still pretty awesome, yeah.
Yeah. But generally, this is what I don't get. But seemingly there's not enough money in there to do this.
So we were talking about golden rice, for example, where you add nutrition to have beta carotenes in the plant, and this is supposed to reduce vitamin A deficiency in the population. And apparently the development was too expensive for the companies, which is why it's now a public company that's doing that in the Philippines. And they're being attacked by farmers, but that's another thing.
What's going to be on the next episode, Dennis?
For the next episode, I talk with Nuno Henrique Franco. For those following our rotating curation account on Twitter, @SFPRokur, the name may ring a bell. He is a researcher for animal well-being, and we talked about why we use animals in life science, the hurdles researchers have to jump in order to be allowed to do animal research in the European Union, and some ethics. If you like our podcast, don't forget to subscribe on the podcast app of your choice. Give us that sweet 5-star rating and check out our Patreon community on www.patreon.com/sci4progress.
Goodbye, listener.
Bye-bye.